Hi, this is Miss Litton, and this is my wonderful Period 1 APBio class. Say hi. Hi. All right, so we just got a little bit farther than this, so let's go over and review this one more time before we start. Here comes Mr. Happy Sperm. He is haploid. The egg is haploid, and sitting on the sperm's head is a cap called an acrosome. Inside that acrosome, there are, starts with an E, enzymes to help you digest the summer concert tour. Zona pellucida. Boy, that was weak. Okay, so you're going to look over that some more. I know you don't have a test or quiz on it, but you need to start getting these. To digest the zona pellucida, then the two haploids will become a diploid zygote. Good. That diploid zygote will do cleavage, and form a solid ball of cells called a what? Morula. A solid ball of cells called a morula. Then you're going to pump in some salt, right? And who's going to follow it? Water. And you're going to form a blastula. And what is a blastula? A hollow ball of cells. Okay? And at this point, we just have one layer. And like a red rubber playground ball, if you could punch that in, that red rubber playground ball, you would form and let out the gas. You can form a gastrula, which is a layered ball of cells. Initially, there are only two layers. The ectoderm, which is the outside, and the endoderm, which is the inside. Now, the way the mesoderm originates depends if you're a protostome or a deuterostome. And let me tell you where that comes from. When it invaginates, this cavity right here that we had in the blastula, that was called the blastocele inside. The blastocele is going bye-bye, okay? And a new cavity is forming. And the new cavity is called it could be the archenteron, or it will eventually become the gut. And archenteron, that's what I always say, but other people say archenteron. It's debated. Here's the blastocele, it goes bye-bye. There's going to be an inner cell mass in there, but inside the gastrula right here, this is going to be the gut tube. And your gut tube, there's an indoor and an outdoor. What's the indoor for our gut tube? The mouth. And what's the outdoor? Anus. And the opening right here to the archenteron is called the blastopore. And if you're a protostome, then the blastopore will become a mouth. And these are things like spiders and worms and mollusks. If you're a deuterostome like us, that blastopore is going to become our anus. Okay, so you're sitting on your blastopore right now. Okay, and then you have these two layers originally, and I don't know if you can see down here, it kind of looks like little Mickey Mouse ears showing up here in pink. Okay, those are the beginnings of the middle layer, which is called the mesoderm. And we get our mesoderm. You can get it from outpocketing of the endoderm. You don't have to learn all of that right now, but I want you to look right here. Here you can see the gastrulation part, and when it folds in like this, if there's no yolk, it's a real easy fold like this to get your two layers. But depending on how much yolk you have, depends on how difficult that will be. If you look here, you have a frog cell, and what happens, they just can't fold in like invagination like this. It has to grow up and around the yolk that's in the egg. So there's a movement. And then here, this is a chick. They have so much yolk, they can't even cleave it. So that happens on this blastodisc right on top of the yolk. So you don't get that movement like you see here, real easy movement. More movement, easier movement when there's less yolk. So here's your gastrula. And then your nerula, this will be the beginning of your nervous system. And what you see right here is you can see in the frog, you can see the blue is the ectoderm, the yellow is the endoderm, the pink is the mesoderm, and this pink area right here in the top, if you can see, there's what's called a notochord, and the notochord induces the tissue above it, and we're going to talk about this term induction. Do you remember it from your Bozeman? A little bit. So it influences, it's like peer pressure on the other tissues. It gets the tissue above to do invagination and form a spinal cord. Now, for us, the notochord eventually becomes our vertebrae that protects our spinal cord, but it depends on what kind of organism you are. But here's the notochord, This little groove right here is going to be the spinal cord, the dorsal hollow tubule nerve cord. And, okay, take a look right here. So you can see that neural tube, you can see the notochord, you can see the endoderm. Now, to learn what each of those layers is going to become, if you think about your endoderm, okay, your innermost layer, your gut tube, okay, what are the two systems you can get to going through our mouth? Digestive and respiratory. So the digestive tract and the respiratory tract come from the endoderm and any organs associated with them, like the lungs or the liver. Okay, the ectoderm, what's our outermost layer? Skin. And then what can I feel right here? And what you just saw invaginating in, that's your, what system? I can feel. Okay, I can feel my skin right here. What allows me? What just folded in?
Oh,
nervous system. Okay? So, it just folded in. That neural tube I made. Okay, good. I was waiting for that explanation because I wasn't sure, but that's good. Okay, so the nervous system and the skin, and then a few like odds and ends, like the lens of your eye also comes from the ectoderm. And if you remember the endoderm ones and the ectoderm ones, because there aren't that many, then whatever's left over comes from the mesoderm. Okay? So I want you to review those. I'll pull them out. Oldest bio buddy, you can go first, and you can talk about the ectoderm. Go ahead, oldest bio buddy.
The ectoderm is
Nervous.
nervous system
Okay, youngest bio buddy, you do the endoderm. Which one?
It's so weird.
Do you like the word mucus? And oldest bio buddy, you take care of the mesoderm.
Mesoderm is like musculoskeletal.
Has anybody noticed that the words match the colors? I had to take time to do that.
I appreciate it. It really helps.
All right. Now, you have charts in your book where it spells everything out and what comes from it. But if you can just generally think about it, okay, ectoderm outside, skin, right, epidermis, and I can feel this skin. And I know through that invagination to form the neural tube that my nervous system has to come from the ectoderm. Endoderm, my innermost, my gut tube that was originally the archentron. What's going to come from that? What two systems? Digestive and respiratory and any associated glands and organs. And then the middle layer is the mesoderm, and it'll just be everything else. Okay. On your notes, let's fill this in, I think. Yes? Not it. Whoever won the not it, just ask 2 quiz questions while I open up the noteboard. Layers? All right, so we have details of human fertilization done. Embryonic development, you have the cellular stages of development, the morula and the blastula. You have that filled in? Tissue stage of development, the gastrula, what do you have there? Layer bulb cells, good. Then you see the layers. Ectoderm, outer layer will form what system? Nervous system, epidermis of the eye. You have everything from mesoderm. Endoderm, what you just need associated glands. And then the archentron is the new inner cavity. The blastocele is gone and it will become your gut tube. The archentron will be your gut tube. The blastopore is the opening to the archentron, and it'll either become your or What could it become? Mouth or anus. Mouth or anus, depending on what kind of organism you are. Okay, the organ stage of development, the neurula, when the nervous system forms from the ectoderm, you have first a neural plate and then a neural tube. First you have a neural plate. See that right here. Okay, so here's your neural plate. And remember, it's this notochord from the mesoderm that's inducing that to happen. You have a neural groove, and then you have that neural tube. The anterior end will become the brain. The rest will become the spinal cord. Okay, in B, it was induced to form by the notochord. And the notochord will become the vertebral column in vertebrates. And now let's talk about some somites. Okay? So I want you to think about this. Sperm fertilizes an egg, zygote, how many cells? 1, right? And then you're 2, and then you're 4. Okay, so it keeps doubling up. And as this is starting to make more and more cells, these cells are going to start to differentiate into specific tissues. And we're going to talk about how does this know to go be muscle, or this knows to go be brain. How does it know that when you're just this ball of cells? So when we start to talk about that process, we're going to talk about segmentation, and that there's differences between the front and the back of you, right? Okay, and there's differences as you move from the anterior to the posterior end, and we'll talk about the differences there. But part of that segmentation along that neural tube is you start to have these precursor cells, and depending on what genes are activated, will then get you different proteins, which then give you different structures. So ultimately, we're going to look, what is the differentiation along what causes that? So that different genes are activated in different locations. So that's part of your somites. And I have midline mesodermal tissue that does not become notochord, 2 longitudinal masses of tissues, and they become these somites. Somites will become muscles associated with the axial skeleton. And then dash segmentation. Segmentation. Okay, so when you look along the vertebral column, you can see how it's differentiated out into those little squares right there. Those little squares are somites, and those somites are specialized to differentiate into a particular tissue. And I might have to remove some students. Do I? No. What did we say that there was a new list we were going to be?
Brown girls.
What was it?
It was things brown people do.
Oh, yeah. Maybe we shouldn't do that. No. That's not what we're going to do. Let's be our favorite ice cream today. Chocolate, baby. Favorite ice cream. Cookie, anything? Oh, favorite ice cream in the freezer. That all ice creams.
Ooh, I like
Vanilla. 2 vanillas. That's my son, my oldest son. What kind of ice cream you want? Vanilla. Want anything? No, vanilla. He is so plain.
Oh, Jesus.
The numbers are going to come.
Don't tell me exactly.
I'm going to pause you for a minute. Okay, let's go over this. 16 of you got this right. Okay, put the following in the correct order. Cleavage first. Please tell me what cleavage is in our lesson.
A little like
Repeated what? Cell division. Without any growth. Without any growth, okay? And then marula. Describe a marula. That was next. It's a solid ball of cells. Okay, 5 was your blastula. What's your blastula? Hollow ball of cells. What's the cavity inside the blastula called?
Blastocele.
Blastocele. Good. And then you said gastrula, and gastrula is a
Full layer.
layered ball of cells. The outermost layer is the, look at me, focus. What's the outermost layer called? Ectoderm. What will the ectoderm be?
Skin,
Nervous system, lens of the eye. What will the endoderm be? Digestive and respiratory tract and any associated organs and glands, and then the mesoderm will be everything else. Good. Okay. So gastrula, and then your nerula would be next on here. Where are we at? 5. Blastula, gastrula. 4. Nerula is the beginning of your organ system, specifically your nervous system, and in order to get the tissue to form, your spinal cord. Where did your spinal cord come from? The ectoderm. And what got it to fold in like that? The notochord. The notochord. Good. All right. Next question says, which of these stages is mismatched? Only 13 of you got that. The blastula is not the beginning of the gut formation, because the blastula is just a what? Hollow ball of cells. You don't get the gut formation until you're at what stage? Gastrula. Gastrula is when you form the gut. 8 of you said that C was wrong. The gastrula is by definition a layered ball of cells. So that's why, if the only thing I can think of that might have thrown you off is you were thinking originally it's 2 germ layers, but by the time you're at you're done with gastrulation, you're at 3 germ layers. So make sure you know that. Check your BioBuddy screen right now, see if they got that one right or they need a little assistance. Ask them if they need a little assistance. Okay, so in some animals, the notochord is replaced by the vertebral column. The notochord does become the vertebral column. It does not become the neural plate. The neural plate is the beginnings of the ectoderm, which is then going to be the neural tube. Okay, the notochord is below that. Those are 2 different things. Check your BioBuddy screen, see if they were one of the 6 that struggled. Come alongside them. Look at their screen. Okay, and the archentron will develop, we talked about this a couple of different times, the gut. Okay, the archentron will develop into the gut. All right, I'm going to take a pause right here for a minute. All right, so our next topic is developmental processes, and developmental processes requires 3 different items here. At least one of them I know you're very familiar with, right? Okay, what's apoptosis? Programmed cell death, purposeful death, right? So an example of that would be the cells that die in between the digits of your fingers. So we don't have pads for hands, we don't have paddles, okay? We have fingers that move around. So cells had to die in between that. When your neurons are extending out of your brain and trying to find target tissues, some of them win, some of them get a target tissue, and some of them don't. Those die. Between your radius and your ulna, the cells died in between, allows the radius to cross over your ulna, so you have this type of movement. If that cell death did not happen, we'd be like, hi, friend. Okay? So we wouldn't have any of this rotational movement. So apoptosis is a normal part. Cellular differentiation, if cells are differentiating, they're becoming, what would be a good word? It starts with the S. Specialized, specific, I like all of that. Morphogenesis has to do with tissues moving, okay? So development requires all 3 of those. So on your notes on the introduction, development requires 3 interconnected processes. Cellular differentiation, which is cell specialization in structure and function. Morphogenesis, which produces shape and form of body, and later pattern formation. And then apoptosis, which is programmed cell death. Here we go. All right, so here you can see a little embryo becoming specialized and differentiating. So let's talk about how this happens. Get this in your head. Every cell has the exact same DNA, right? So if every cell has the exact same DNA, why are cells different?
Oh, different parts of DNA.
Different parts of the DNA are expressed. Exactly. So why is it in these cells this DNA is expressed, and over here in these cells this DNA is expressed? That's what we're trying to understand. So when you have the exact same DNA and you have full developmental potential, that's called being totipotent, full developmental potential. Now, if you look at this cell right here, you can see the nucleus in here, and you can see red and green, and these are called maternal determinants. The reason why they're called maternal is the sperm doesn't contribute much other than its haploid nucleus. All the cytosol, all the proteins in the cytoplasm, the cytosol is just a fluid portion of it, all of that comes from the mother, right? All the organelles come from the mother. Do any other organelles besides the nucleus have DNA in them? Mitochondria, boom shakalaka. All of your mitochondrial DNA came from your mom. And they can look at people's mitochondrial DNA and trace lineage, trace ancestry through the because that's your mother's line, okay? So these maternal determinants, you can see there's red and green unless you're colorblind, okay? After the very first division, the very first cleavage, which one, the top one or the bottom one, has more red?
Bottom.
Okay, so the bottom one here you're saying has more red in it than the top one. Now, if these are receptors, right, internal receptors for a hormone or something, right, if these are transcription factors themselves, do you see how that can influence what's going on in the nucleus? Right? Because we know about the process of signal transduction, where something's received, right? And then it can influence the behavior of that cell. So right now, you have different things in the cytoplasm that's going to affect that DNA. If you look here at the next division that takes place, which, if you look at the top, this one up here has 3 reds, this one has 3 reds, this one has 1, 2, 3, 4, 5, 6, this one has 1, 2, 3, 4, 5, 6, 7, 8. So this one has more red than any of the other ones. So how that influences this DNA would be different. And this is a very simplified drawing. This actually happens when you look at, if this is an egg and the sperm fertilizes the egg, the point of where the sperm hits the egg, that will be the anterior region of the organism. If you touch the top of your head right here, this is where your father's sperm penetrated your mother's egg. Oh, that was hilarious. She's all, no. She's like reaching to touch the top of her head. All of a sudden, the top of her head. No. Gross. Okay? So that becomes the anterior regions because what's in the cytoplasm right there is slightly different at this end than it is at this end. Where you have more yolk here at the bottom, that will be the ventral region, or called the vegetal pole in the frog, and the opposite region will be the dorsal region. So if I drew this out like this, okay, so here you have the sperm where it's going to penetrate the egg. This region right here has sperm, this region has no sperm. This region down here, has yolk, and the area above it has no yolk. So we could break this into 4 quadrants right now, okay? This one has sperm. This one has sperm and what? Yolk. This one has no sperm, so it just has yolk, and this one has nothing. So right away, we have 4 quadrants right here. So each of the nuclei in each of these cells is going to be exposed to something a little bit different. And then that causes another slight gradient when this cell divides, and another slight gradient because this one has a little less of the sperm, right? And it's not so much the sperm, but any chemicals that came in with it. And this is what begins that process of differentiation. And it starts our anterior, posterior differentiation, right? Our dorsal and ventral differentiation from the very first few divisions. And they have actually done, you want that? They have actually done experiments on this with frogs. And so this is normal frog development. This is a full, has full developmental potential. And this is after the very first cleavage. And if they separated, now normally these wouldn't get separated, they would just develop, right? If you separate them out, they each develop into a normal tadpole. But this one over here, they caused the cleavage to be in this region at a different spot. And when they pulled them apart, this one right here was had full developmental potential, but this one did not. So if you look, this part right here is called the gray crescent, and you can see it in frogs. And if they both got a piece of the gray crescent and then you pulled them apart, they were fine. This one got all the gray crescent, he developed normally. This one got no gray crescent. He never went through the process of gastrulation. And what they've come to find out is it's that where the gray crescent is going to be the dorsal lip of the blastopore. It's where it's going to start to do the infolding to go on to the next stage. No gray crescent, you don't move on. Okay, so what's in that cytoplasm at that region does affect development. Okay, go ahead. What were you gonna?
Could this happen like humans?
Yeah, but it's hard to do these kind of studies on humans because the whole life thing. Yeah, it's tricksy that way. All right. So if you go to the notes on number 1, okay, our underneath cellular differentiation, every cell contains the exact same what? DNA. And our totipotent. Number 1 is cytoplasmic segregation. And I said A about sperm penetration. That will be the anterior end. The other one will become, oh, that's okay. That's what I meant. Sperm penetration is the anterior end, and it will become the anterior end of the organism. The opposite one will become the posterior end. Poles. The vegetal area is the area of yolk. It's vegetal, not vegetable. Okay. The animal pole will become the dorsal region. So this is anterior. This is posterior. Where you have yolk, this is the ventral region called the vegetal pole. And this is the dorsal. This will become this is the animal pole. Okay. Another picture. Okay. And then cleavage, cytokinesis will separate the ooplasmic female proteins, the maternal determinants, which will activate different genes, which will result in differentiation. Youngest bio buddy, IYOW, right here, go. In your own words.
Wait, what? Stop. I'm going.
All right. Now, let's take a look at this. Here you can see the sperm fertilizing the egg. Here's a marula. You know what a marula is? What's a marula? Solid bulb cells. Good. Solid bulb cells. And it's totipotent. What does that mean? Full developmental potential. Okay? Now, you're at the blastocyst or a blastula. And you remember I told you in humans, inside the blastula will be an ICM, inner cell mass. Okay? This inner cell mass right here, this blastocyst is going to be different structures. It doesn't have full developmental potential. Okay, so being totipotent, are there terms there I need you to write? Totipotent gives rise to all the cells. The totipotent marula gives rise to all the cells that make up the embryo and support the tissues as well. So it's everything. You can create a whole new organism from that. But now this pluripotent, the inner cell mass, the inner cell, oh, that's funny, I fixed that, but I went back to it again. Okay, the inner cell mass gives rise to all the cells of just the embryo. So you can't get another embryo from these cells right here. They've already started down the pathway of differentiation. The embryo is only going to come from the pluripotent cells of that inner cell mass. And now here, they're starting to specialize. Uni, what does uni mean? One, right? So when you're unipotent, you can develop along only one line. So totipotent, pluripotent, unipotent. And so the word, what's the word you put at the end of pluripotent? Embryo, good. What'd you put for unipotent? One, good. All right, and now this part you have already learned about, and this has to do with induction. And this is when one tissue influences another tissue. It's influencing its morphogenesis. It's influencing its differentiation, okay, when it does this. So here this notochord is influencing, and the notochord is mesodermal tissue, is influencing influencing. That's a new word. I'm making you more rich. I'm affluencing you. Influencing the tissue above it to form the neural plate, the neural groove, and eventually that neural tube. And also they've done this with experiments with frogs and eyes. They've gotten the tissue. There's something called the optic vesicle that gets the tissue above it to enfold. The optic vesicle does that to form the lens. And then the lens gets the optic vesicle to become the optic cup, which becomes your rods and cones of your retina. So it's like it influences this tissue, and then this tissue develops it, influences it right back, and then influences it again. So they're kind of tag teaming each other. And what they did is they took optic vesicles and put them along the legs of frogs, and got eyes to develop down their legs. Now, these were not functional eyes, and the reason is because you didn't have the neurons traveling to those areas in order to anchor them and make them useful where they could look around. But they got the structure of the eyes to form. And so you can override what that tissue is supposed to be doing with that peer pressure of induction, because you're saying, I want you to go be this. They've actually moved notochord tissue and added it to salamanders, embryonic salamanders. They've removed a notochord from another salamander and added it to another salamander, and they got it to develop two heads, two spinal cords as a result of that. So they're saying, no, you will become this tissue. So they override the development. And there are some examples here where they're doing that same thing, where they're moving the tissue, and so they got two neural tubes to fold, to become as a result of that. Okay, one organism that's been studied a lot is a nematode, and this is a pseudocoelomated worm here, and he's always 959 cells. And they have been able to trace where all 959 cells originated from. And its name is C. elegans, and it takes about 3 days to develop. And they can tell at each stage of development, oh, it's going to go form these cells, it's going to go form these cells, it's going to go form these cells. So it's been studied quite a bit. All right, so let's see what else. Induction is when one tissue influences the development of another, either by direct contact or release of chemicals or morphogens. It seems to activate certain genes by generating transcription factors. Blue, tell Slate what a transcription factor is. Okay. Your examples: the gray crescent in the frog became the dorsal lip of the blastopore. It's called the primary organizer. It must have this in order to develop normally to form the gastrula. So that's what I was showing you back here. Let me just reiterate so you know where I'm at. You needed this gray crescent to influence the process of forming a gastrula. And then your other example here is the notochord induces brain development. And you already have optic vesicles induce the formation of the lens. And then you have the worms. Slate, tell them 2 things about cellular differentiation. Slate, tell them 2 things you know about cellular differentiation.
So it's influenced by the different, like how specialization. Yes. How they activate certain
Okay, here we go. Okay, so morphogenesis. Okay, so now we understand a little bit about cellular specialization, though we're going to hit this again in just a moment. Morphogenesis is how cells move around. Like, for instance, your eyes start up right up here, and then they migrate like this, down to these forward-facing eyes, okay? So morphogenesis is movement of cells that changes the shape and form of the body parts, becoming tissues, organs, etc. So pattern formation, cells divide and differentiate, enable the process of morphogenesis. Okay, so what influences development? We already talked about cytoplasmic segregation, those maternal determinants. What's in the cytoplasm of the cells is different, so it's going to influence the DNA. Induction we've already talked about, when one tissue influences another. And this is how it's working, okay? So some sort of, because what's in the cytoplasm is different, it's influencing certain genes. Those genes then, when it's transcribed and translated, they'll make another protein, which then activates other genes, which then activates another one, and so on and so on. And they can be in a gradient as they go down through the organism, and it causes then ultimately this segmentation. Okay, so blue, you explain morphogenesis, and slate, you say what a morphogen is. Differentiate between those two. Go ahead. Okay, now, got the difference here? Okay. Yeah, morphogenesis is the process, but morphogens are the things that are actually triggering different genes. And so what ends up happening is you have a difference just right here, and let me simplify it because I'll try this a couple different ways. But if you have a developing organism, okay, and if you have a chemical gradient and you have this that's less and less here, and you have another gradient here, you could have a ratio, right, between the red and the, between the green and the blue that's going to be a little bit different, how much green you have to how much blue at every single point. And so it could be at a certain inflection point where you have this, you get another chemical that's produced. And over here you get a different chemical that's produced. And now you have a ratio between the green and the blue and the black and the green, which then induces another ratio to be here, another one over here, and so on and so on and so on, because there's just these slight differences in the chemicals which affects the DNA slightly, which then gives you these proteins in a gradient, which then begins the process of segmentation. Okay? And if you look, putting it all together, okay, so this is real simple where it's just red and blue, and there's that slight difference between that, which is then influencing other genes, which starts to separate them out. Now, the genes that are really important are called homeotic genes. And these homeotic genes are eventually producing the transcription factors that influence which genes are transcribed and which proteins you have. All just a difference of a gradient. And so take a look right here. Okay, so you have a morphogen. This morphogen affects this DNA right here. It's coding for then another morphogen, which then codes for a different morphogen. And I keep telling you the same story, but it's just slight differences in which genes are turned on at which time. And here, okay, take a look at this picture. So here's this specific sequence of DNA that is coding for this particular protein. This specific piece of DNA that codes for that developmental morphogen is called, these are called homeotic genes. Blue, this is you. Go ahead, explain the bottom part underneath the diagram. Okay, so what's inside of your homeotic gene? There's another one now, the homeo box. And the box is the one that's actually coding for the transcription factor. Okay, which is then going to influence the next step. Okay? Yeah, it keeps kind of breaking it down. But anyway, on your notes, go into under morphogenesis. We have B. It says pattern formation. There's nothing you have to write in there. I just want you to see it again. Under described B, pattern formation. Cells divide and differentiate. They enable the process of morphogenesis. C, though there are so many different types of animals, they share common sets of genes. They share common sets of genes that direct pattern formation. Okay, and then looking at Drosophila, anterior posterior axes, the first step in pattern formation results from maternal determinants. Okay, what does that mean in parentheses? What do you think that means? mRNAs, what could that be? Messenger RNA. So they have different ones, right? Okay, and then chemicals and morphogens in a gradient. You don't have to memorize bicoid and nanos. I have that in parentheses, so if you want to make that small, you can. Underneath segmentation patterns, there are gap genes that are temporarily activated by the morphogens. I have Kleenex, baby. And pair rule genes, genes that code for preliminary segmentation, which in turn activate segment polarity genes. And so it continues. So I'm not going to quiz you between gap genes and pair rule genes. I just want you to see that there are different genes influenced at different times. But I do want you to know homeotic genes, often called selector genes because they select for segmental identity, dictating body parts. Dictating body parts. Okay? And they are highly conserved. So it doesn't matter if you're looking at a mouse or an embryo, they have similar series of genes that code for their anterior end, which they colored here in green. And then they have similar genes here. And this is a mammal versus a fly. They have these same pattern formations developing this pathway of development. So highly conserved, meaning that they are found in many animal genomes. And C, Blue, you can do that one. Do 3C. We already talked about it. Yes. Yes, we can. So what they did here is there's something, in this part you don't need to know, but they're called imaginal discs. Remember those somites I showed you about in vertebrates that directed development of our muscles and bones? In insects, they have these things called imaginal discs, which are going to develop into antenna or legs. And so what they did is they moved some around. They took imaginal discs from one and they doubled them up right here. And so as a result, those imaginal discs, with the ratio of chemicals they had in their cytoplasm, those morphogens influence then double wing formation right here. Or they've taken imaginal discs and of legs and switched them with antennas so that they had legs growing out of their heads and antennas here out of the sides. And so because it sends them down that developmental pathway. It doesn't matter that it's in this place and I'm supposed to have a leg here. Okay, it doesn't matter. It's whatever DNA is activated, that's going to develop. They've done this with chickens where they've got them to develop a set of wings and then another set of wings right on top of it. So they have double wings that don't function well. Okay, but they're showing that whatever genes you turn on, that's what you're going to have in that particular location. All right, and then the last part about apoptosis, programmed cell death is needed for normal development of the eye, the nervous system, and phalanges. So I know that's a lot of different things, so I wanted to kind of give you an idea, something to kind of help you possibly with it. So we're trying to get development to happen. That's our goal. So if you're playing pool, you're trying to get all the balls to go into the what? The pockets, right? Okay. So that's our development. Playing the game of pool, getting development to go along, that's like morphogenesis. And the pool cue, the stick, it hits the ball, which is white, which impacts other colored balls. That's an example of induction, where one tissue influences another. Just like you can hit the white ball and it influences all the other balls. Okay? That stick is the inducing tissue. The cue ball, the white ball, are morphogens. You're using morphogens to influence the other tissues in their development, in their movement. Those morphogens are transcription factors. Those transcription factors were coded by homeobox genes that were sitting in homeodomains, right? So influencing, then what happens is when I use this white ball to hit this black ball, right, that can then influence another cell, which then can influence another cell, right? And so on and so on. And that gets the ball rolling in development, okay? Let's ask a few questions. Yeah, you can. You want to be yourselves? I won't know who you are, so it's okay. I won't call you out if you miss this.
What do we Jake?
BJ. No, I'm Jake.
Yeah.
Yes, yes. Hurry, we're answering questions. Guys, if you survived that, that was the hardest part of today. Literally, the rest of it's very easy. You should. Now we just get to talk about this pregnant Hawaiian that's coming up. She's married for many years. It's very important.
An important fact. Life lessons with this,
What? Yes. She's in a stable, loving relationship with her partner, which I don't care who her partner is, but it's like she's in a stable and loving relationship. Raising her child. Let's go. 3 questions. Are they hard? All right, guys, we're going through this because I can't wait longer. Okay. Are you logged in twice? Is that why I have so many people who haven't answered? Did you re-log in as somebody else? That's what it is. Okay. What? Okay. So 4 of you got this right. Morphogenesis is associated with proteins in a gradient. Right? We've talked about that. Proteins in a gradient. Induction, when one tissue influences another tissue. Transcription factors affect what DNA is transcribed and what proteins you get. And homeotic genes, it's the homeotic genes that are coding for those transcription factors. Ask your bio buddy, because there are only 4 of you that found the correct answer there. What did you leave out? And do you understand it now? Check with your bio buddy. I only have 4. All right, 10 of you got this right. Which of the following one is mismatched? You may not know any of these, but you know that the lens comes from the ectoderm, right? 9 of you said brain and ectoderm. Where does our nervous system come from? The ectoderm. And our brain is part of the nervous system, okay? 3 of you said gut, endoderm. Our gut tube comes from that innermost layer. It was the archenteron, right? And then it becomes our gut, and the opening, the blastopore, is either going to become the mouth or the anus. For us, it's the anus. And those are the only wrong ones you picked. And this one, the ability of one embryonic tissue to influence the growth and development of another is termed induction. Now let me help you on the answers you missed. The process of one tissue influencing another is called induction. What do they influence with? A morphogen, which isn't in here, okay? Morphogenesis is what happens after you've influenced them. Do you see that? Okay, morphogenesis is the end product, right? The development that we're trying to get it. But the ability to apply peer pressure, one cell to another, that's induction. What do they apply that peer pressure with? They apply it with morphogens. What process do they get to happen? Morphogenesis. Okay, question, Jake?
Well, it was like, I was just a little confused with number 2 because I thought like.
Okay, wait, any other questions on 3? Okay, so I will go back to 2. Go ahead.
So I know you said like ectoderm, you said earlier, I know it's the nervous system, and I get that, but like, isn't like the outermost layer, and I don't see how like the brain is the outermost layer.
No, sweetheart, the brain in your body is not right now the outermost layer. I just told you that your brain came from the ectoderm invaginating because the notochord told the ectoderm to invaginate, and that became your neural tube. So that's where the layers, what they became. So that's not like, yeah, your skin's on the outermost layer, so that's easy to remember coming from the ectoderm. You just have to learn that the nervous tissue came from the ectoderm folding in, and you got your neural tube, and then at the anterior end, a brain developed. Okay? It's just like you just got to know that. All right, ask your bio buddy any questions, concerns on those 3 questions that they want to go over. Okay, let's see our pregnant Hawaiian. She's married in a stable and loving relationship. Okay, now, first thing I want to talk about are mammal membranes. And you can see on that chart, I made that chart what? Can you tell I made it tiny? It's supposed to be tiny. Okay, but what I want you to see, here's the big picture that I want you to see, is similarities that you can see between a reptile and bird and a mammal. Okay, so for instance, you have the amnion. The amnion is the one, that's the membrane that's right around the developing embryo, and we have an amnion that's right around our developing embryos too. So that's a similarity here in development. You can see the chorion, okay? The chorion, if you've ever hard-boiled an egg, and you crack that egg and you peel off the hard shell, you know that membrane that's right on top of the white that's kind of, you can move it? That's the chorion. That chorion is where all gas exchange takes place, okay, for the chick when it's developing. For us, our gas exchange is going to take place right here where we're attached into the placenta. So our outermost layer is buried in with the placenta. And what you have in here, in this section, I just want to point out, is the blood pathway between the baby and the mother. It's a what kind of situation? You've seen it before. Countercurrent exchange. Yeah, between the mother and the baby. So I just wanted you to see some similarities between the membranes of like a chick or a reptile and a human. As far as embryonic development goes, most of us have heard of the trimesters before, so I just want to start off with that. In your first trimester, usually you can't tell somebody's pregnant, very early development. And by the way, if you're going to drink a bunch of beer, the time here in the first trimester is when it has the greatest influence on the developing embryo, when it's just a few cells. But if somebody walked up and they were in their first trimester and they were at a bar and they were not, you wouldn't even know they were pregnant. At the third trimester is when it would have the least amount of effect. Do I think I want you to go get drunk when you're in your third trimester? No, but if you saw a woman who was super pregnant and she's waddling up to a bar and knocking, you'd be like, what's she doing with the baby? Okay? But when she has the greatest effect is early on. Why am I telling you this? Because I don't want whoops and I slipped and fell on a penis, and you want to do that purposely, okay? And you want to know if there's a possibility you could become pregnant, because if you are not paying attention to what you're doing with your reproductive organs and you potentially have made a baby within you that you're going to plan on having and raising, not even when you even know you're pregnant, you know, any one of you right now, any one of us in this room could be pregnant right now and we wouldn't know if you're in the first trimester, okay? Except for, okay, good job. Varsity. Oh. Okay. You wouldn't know, okay, because you don't see it, but that's when you can have the biggest damage. Second trimester, you're starting to show a little bit more and you have movements, and then the third trimester, you notice the baby turns around because they head first when they head out. Okay? Now, here is development fast forward. Okay, this is your summary picture to walk away. This is the end of the story. This is the conclusion I'm giving you right now at the beginning, but I'm going to show you pictures. W stands for week 1. And in week 1, you get to the blastula stage and you begin to implant in the uterus. And I always think blast into, the first week. And you get to the blastula. Okay? Week 2 is the gastrula stage in development. And the way I remember this is if you go number 2, you might have a little gastrula. Okay? Gas. I know it's gross, but you'll always remember it. Week 3 is the nerula stage and the heart pumps. And the way I remember that is if you take this 3, okay, take this 3, yes, exactly, and you turn it sideways, that would be a cursive N for nerula, and you can turn it into a heart. Okay, week 3, the nerula and the heart begins to pump. Okay, weeks 4 and 5, which is in month 1, you have limb buds and your senses. You have more senses than that. I'm just putting this on here. Week 4, how many limbs do you have? 4 limbs. Okay, and you're probably familiar with 5 senses, but there are more than that. Okay, so limb buds and senses by weeks 6 through 8 in month 2, the baby starts to show reflexes. They respond to large, to sound. And then week 10 is when you get the placenta. And by month 3, you can see genitals. And by month 4, you can hear the heartbeat. And the way I remember that, month 4, so they're farther along, how many chambers do we have in our heart? 4. Not to be confused with week 4. But week 4 is really early on. As far as how it looks, this is an early embryo. These are the eyes that have been already formed. This is the yolk sac that provides nutrients until it gets nutrients from the placenta. Okay, this would be following conception. That might be a solid ball of cells. What would we call that? A marula. Okay. Now, that inner cell mass that I talked to you about, you guys, there's Kleenex.
It's empty. Yeah.
Oh, look at the coverage.
Jeremy, where do you go?
Ouch.
For decoration.
Yeah. So, I'm going to show you a video, guys, up here. I'm going to show you a video, and then this is the beginning of that invagination to form that neural tube. Okay, that's scary. Don't worry about that, but I want you to see up here, look at fertilized egg, blastocyst. Do you know what a blastocyst is? Yes. Okay, here's your inner cell mass. Okay, your inner cell mass, if you see where it's breaking it down into ectoderm, and you can see endoderm. So those should be familiar.
In this video then we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm which travel down the fallopian tube. So here millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring.
That would be like marula.
This is one of the early growth phases.
And then you go into
as
the
the embryo
blastula.
moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal.
Those
And
are
the
the
outer
pluripotent
cells
cells.
give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo.
That's the
Those
neural
lines
tube.
represent sites where cells are migrating in and out.
The brain's going to be
And
at the
here's
far
an
end.
important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes.
Watch.
As development proceeds, there's
tube.
more growth and movement of cells. It'll begin to form a neural tube Here it turns and appendages start to bud out. You see the head forming and the eye, and then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
As you know, a baby. I think he said, as you know, a baby.
Specifically, a young baby.
A young baby as opposed to an old baby? Okay.
Discrimination.
So that little inner cell mass forms that disc. You could see the infolding to form the neural tube. Yes?
So is the outside of the box, is the placenta, what will become the placenta?
Yeah, it will become the placenta and the supporting cells. All right? And then eventually then it's going to differentiate into all those tissues.
So here we're going to go through quickly the early stages of development again. And then we're going to get to this stage when the germ layers are set aside. The three germ layers, the ectoderm, mesoderm, and endoderm. So here you'll see then that the inner cell mass gives rise to the entire adult animal, in this case a human. This same process would occur in a mouse, as we'll see a bit later. If we now look at the embryo after the three germ layers have formed, this video will highlight what comes from the blue germ layer, the ectoderm. You'll see that it makes the nervous system, including the brain, and the skin. The middle part, the mesoderm, green here, gives rise to the muscle, including the kidneys, the heart, and the endoderm gives rise to the whole gut tube. There you see the lung, the liver, the intestine. And to give one more detailed example of this, let's think about the development of the endoderm, and in this case, the formation of the pancreas. So there's the pancreatic bud, which comes out of the endodermal derivative.
Endodermal derivative. Okay. All right. So here you can see, this is month 1 of a human. Okay, and you can see the limb buds, and you can see when we have a tail coming around here. Here's the limb buds, here's the tail. You can see the beginnings of the optic vesicle where it's going to develop into the eye. Month 2, when you start to, this is a 2-month-old fetus, starts to have refluxes. Placenta in the 10th week, and it's called, the chorion is that outer membrane, so it's the chorionic villi, and you have counter current exchange there. And anything the mother has in her body is going to cross that placenta and go into the baby. So if the mom smokes crack, the baby smokes crack, right? Everything crosses through the placenta. I'm going to come back to that one because I want to make sure you finish. So you want to, be very careful what you expose. Any of these things here, you're going to expose your baby to. Genitals by month 3. Month 4, you can hear the heartbeat. Remember we said month 4, 4 chambers? Yes?
Does that mean if you wanted to determine the sex of your baby, you have to wait 2 months for me?
Yeah.
How accurate is that?
Pretty accurate, unless the clitoris, which could become a penis, sometimes that gets confused for a penis. So it depends on how good the imaging is, really, your ultrasound technician and how well they're able to see. And you have to do things like have a full bladder, so you're pushing your uterus up. They're right both times for me. We did about the fourth month. Okay, here's month 5. You can see the umbilical cord. Month 6. Month 7, they're usually starting to turn around now. Month 8. Month 9, obviously this one's outside. Here's a placenta. This is not a human placenta. This is like a goat or something. It's not blue, but you can see the membrane. It's because the animal's dark underneath it, so it looks blue.
Water balloon.
Yeah. Here you can see the umbilical cord that needs to be cut. As soon as you cut it, the baby will look blue for a little bit as they're getting their lungs to take care of the oxygen, and then they'll. Yeah. Here's a cross section of an umbilical cord, the 2 arteries and the vein. So this here is that positive feedback example that we talked about. Get the baby out. You should not push with your face because it's not going to affect your uterus at all, okay? And you can end up breaking some blood vessels and stuff like that in your face. Just pretend like you're having the biggest dump of your life, and that's what it's, how you give birth.
Can you defecate during this process?
You can. I pray to the Lord God in heaven and in my heart that I would not take a dump, and neither time did I. That was a real concern for me. It's like, please, Lord, do not let me dump on the table. And I did not. I did have to go pee, but that happened before the process, and I wanted to just jump up and go pee. And they're like, no, go ahead. And I'm like, and they're like, no, go ahead. Okay. It's like peeing in a wetsuit. And then they're just like, and they clean you up. It's awesome. So stage 1, the amniotic sac's gonna break, out comes the fluid. Stage 2 is the baby, stage 3 is the placenta. Let me give you a few words here to fill that in, okay? There's only a few words. Week 1, unless you already filled it in under embryonic development, is blastula. B, inner cell mass becomes the embryo. Week 2, you should have filled in gastrula. Week 4 and 5, limb, bud, sense organs. Did you already do that? This part that I just showed you, stages, first contractions and amnion breaks. Bye, sweetheart. Second, mother pushes, baby emerges, and third is the afterbirth. And under aging process, the last one, the hypotheses about aging, whole body process, hormonal decline, and extrinsic factors, years of poor health habits contributes most to aging. That was it. All right. Remember, I am not here Friday, and you're going to be reviewing. Hey. Here you go. This is a baby coming out. I just didn't want to leave without you getting that picture. You know, I'd appreciate that.